Hello, I would like to welcome you all to this talk, which will be about my publication with the title Laminin 5:1 stabilizes the pluripotency expression pattern of human embryonic stem cells initially derived on feeder cells. I'm Halima Albalushi, a medical doctor from Sultan Qaboos University in Oman. I'm doing my PhD in Karolinska Institutet under the supervision of Jan Bernd-Steckenberg in Norfertil Research Lab in Stockholm. Before starting to talk about the publication, I would like to start with a brief introduction about the human pluripotent stem cells. Human pluripotent stem cell consists of human embryonic stem cells and human-induced pluripotent stem cells, which hold a great promise for future regenerative medicine. Human embryonic stem cells are derived from human embryo at the stage of blastocyst, specifically from the internal cell mass, and the human-induced pluripotent stem cells derived by reprogramming of adult cells by specific factors. These stem cells are distinguished from other cell types by two important characteristics. First, their ability to renew themselves through cell divisions, even after a long period of inactivity. And under certain physiologic and experimental conditions, they can be induced to become tissue or organ-specific cells with specific function. Human embryonic stem cells with human-induced pluripotent stem cells provide a unique platform to study molecular and cellular mechanisms in humans. They represent an important tool to study early cell development with potential application in the field of regenerative medicine. However, use of human embryonic stem cells and human-induced pluripotent stem cells in the regenerative medicine and possible future use in the clinic require well-defined and controlled culture conditions for these cells. The first human embryonic stem cells was derived on feeders, but the use of human embryonic stem cells in future personalized medicine requires xeno-free and ideally even feeder-free culture conditions. Such xeno-free and feeder-free culture conditions are needed to avoid immunogenicity, microbial and viral contamination, and batch-to-batch variability of the culture matrices used. In the first attempts to create a feeder-free culture system, matrigels, which is a protein mixture of several unknown components and growth factors derived from mouse sarcoma cells, was used. To a large degree, these unknown components and the batch-to-batch variability of matrigel complicate comparability between human embryonic stem cells experiments. In order to avoid variability, we'll divide culture conditions involving, for example, purified matrix protein such as Laminin 5:1 combined with xeno-free media have been designed to further increase the reliability and reproducibility of various differentiation protocols used. The previously described use of human recombinant Laminin 5:1 represents a step forward in generating clinically safe culture conditions. However, evaluation of gene expression profile involving several human embryonic stem cell lines generated on feeder cells and transferred into Laminin 5:1 with special focus on the differences during the first passages and the effects on pluripotency gene expression has not been performed. And that was the aim of our experiment here, is to study the short-term effect of using Laminin 5:1 as a culture matrix to culture human embryonic stem cells initially derived on feeder. The study consists of 3 steps of experiment. In the first step, we transfer 5 human embryonic stem cell lines from feeder to Laminin 5:1, and we assess the pluripotency status of these cells. The second step was to culture these 5 human embryonic stem cell lines on Laminin 5:1 for short terms, and then to analyze the gene expression of these cells. In the third step, we culture 5 human embryonic stem cells on Laminin 5:1, Laminin 1:1, and matrigel, and then we compared the gene expression of these cells. For the first step, we transferred 5 human embryonic stem cell lines from feeder to Laminin 5:1, and we observed that the colonies formed monolayers and continued to grow without any signs of accelerated differentiations in all 5 lines. To confirm the pluripotent nature of the 5 human embryonic stem cell lines we use in this experiment, gene expression analysis of 96 genes using TLDA revealed similar but not identical expression profiles. At passage 4, all 5 cell lines exhibit enhanced expression of 9 genes related to stemness and reduced expression of 4 genes related to differentiation. The pluripotency status and potential influence of the culture method per se on the 5 human embryonic stem cell lines cultured on laminin 5:1 were tested after 9 passages. The morphology of culture cells was not changed compared with the cells cultured up to 4 passages, and all 5 cell lines express a set of 5 commonly known pluripotency genes at the protein level. After passage 9 on laminin 5:1, the ability of the cells to differentiate spontaneously into the 3 germ layers, ectoderm, mesoderm, and endoderm in vitro by the formation of embryoid body was tested. All human embryonic cell lines expressed marker of ectoderm, mesoderm, and endoderm after 28 days of spontaneous differentiation. So from this we conclude that laminin 5:1 provides suitable culture condition for adaptation to feeder and xeno-free condition of human embryonic stem cells derived and cultured on feeder cells. The second step was to culture 5 human embryonic stem cell lines on laminin 5:1 for short term for 4 and 9 passages, and then to analyze the gene expression at different passages. When we compare the gene expression profile of human embryonic stem cell cultures on human feeder and laminin 5:1 at passage 4, we analyzed 17 genes, and we saw that 14 genes showed more than 50% gene expression variation on human feeders compared to 11 genes showing more than 50% gene expression variation on laminin 5:1. While at passage 9 of laminin 5:1 culture, only 7 genes showed more than 50% variation. Homogenization of pluripotency gene expression on all 5 cell lines was observed when comparing expression variation between passage 4 and passage 9. So from this part of the experiments, we concluded that laminin 5:1 has a positive effect in homogenizing pluripotent gene expression profiles between human embryonic stem cell lines cultured for up to 9 passages. The third step in our experiment was to culture 5 human embryonic stem cell lines on laminin 5:1, matrigels, and laminin 1:1, and to compare the gene expression. And that was done to investigate the difference between laminin 5:1 and other matrices used for human embryonic stem cells cell culture. And for this purpose, we culture 2 human embryonic stem cell lines on laminin 5:1, laminin 1:1, and matrigel. We observed a higher incidence of differentiation in cells cultured on matrigels and laminin 1:1 compared to those cultured on laminin 5:1. In addition to that, human embryonic stem cells were attaching and growing in a lower rate on laminin 1:1 compared to laminin 5:1. The differences between laminin 5:1, 1:1, and matrigel became more pronounced with prolonged culture. Although more detailed studies about the long-term effect of culturing human embryonic stem cells on laminin 5:1 is needed to compare them with the short effect presented in our study, we can conclude that laminin 5:1 supports human embryonic stem cell growth and has a positive effect on maintaining the cells in a normal balanced pluripotent state. Similar expression profile of different human embryonic stem cell lines might result in more predictable and therefore controllable behavior of these cells in various differentiation protocols, which is required for the possible use of these cell lines in the field of future regenerative medicine. At the end of this presentation, I would like to thank you for your attention. Also, I would like to thank the co-authors of this work for their contribution. And if you want to know more about my research, you will find me in ResearchGate and LinkedIn. Thank you very much.